Assalamualaikum and hi. My name is Nur Shazana binti Azhari. So now, in this video, me and my team will explain about the Sds-Page. Now, let me start with what is the Sds-Page. Sds-Page is a powerful technique widely used in biochemistry and molecular biology for separating protein based on their molecular weight. Sds-Page stands for sodium hydroxyl sulfate polyacrylamide gel electrophoresis. This method involves the denaturation of protein with Sds, an anionic detergent which impart a negative charge to the protein proportional to their length. The protein are then loaded onto a polyacrylamide gel and subjected to an electric field. Smaller protein migrate faster to the gel, allowing the separations based on size. Understanding Sds-Page is very important for protein analysis, including purity checks and molecular weight determination.
And today, I will explain about the importance of Sds-Page in analytical biochemistry. Sds-Page is a crucial technique for protein purification and characterization. It will allow us to separate proteins based on their molecular weight and will aid in molecular cloning and expression studies. And in clinical diagnostic, Sds-Page is used to identify and analyze protein abnormalities. And additionally, it plays a vital role in proteomics by helping us understand the protein composition of cells and tissues.
The principle states that molecules separated based on their electrophoretic mobility will migrate towards their respective electrodes when placed in an electric field. Electrophoretic mobility depends on the molecule structure, shape, and charge. The structure and charge of the proteins are mainly responsible for the migration. The smaller molecules will move faster due to less hydrogens, whereas larger will move slower due to greater hydrogens. Thus, molecules are only separated based on their molecular weight.
Additionally, there are other common methods used in Sds-Page. These include Sds-Page is used in combination with the Western blotting for the determination of the presence of specific protein in a mixture of proteins, or for the analysis of post-translational modification. Next, native page is another method for non-denaturing process for analyzing protein complex. Other than that, 2D Sds-Page is used to separate protein based on the two properties, isoelectric point and molecular weight.
These are the list of instruments involved in Sds-Page: gel casting stand, glass plates, spacers, comb, electrophoresis tank, power supply, electrodes, micropipettes and tips, UV or visible light transilluminator, gel drying apparatus, and imaging system.
The material used for Sds-Page including acrylamide and bisacrylamide for forming the polyacrylamide gel matrix, SAS for denaturing protein and providing them with a uniform negative charge, APS for initiating the polymerization of acrylamide, TMAT for catalyzing the polymerization process, trace buffer for maintaining a stable pH during the gel formation and electrophoresis, trace glycine SDS buffer for providing ion that carry the current and maintaining pH during electrophoresis, sample buffer containing SDS, bromophenol blue, glycerol, and a reducing agent to break disulfide bonds, staining solution either Comasi blue stain, silver stain, or peach blue stain for visualizing protein after electrophoresis, destaining solution to remove excess staining solution, and lastly, distilled water for preparing solution and washing step.
Now, I will proceed with the type of sample. Sample choice is very important for accurate result. Common sample include cell lysate, which contain a mix of protein from cell or tissue, useful for studying protein profile. Purified protein, often specific analysis for precise molecular weight and functional studies. Complex mixture, like serum or cell culture media, provide insight into protein interaction.
For sample preparation, a loading buffer containing SDS, beta-mercaptol ethanol, bromophenol blue, and glycerol is added to the protein samples.
Protein samples may be biologically derived from examples from prokaryotic or eukaryotic cell tissues, viruses, environmental samples or purified protein, and when the sample buffer has been added, the samples are then heated at 95 degrees Celsius for 5 minutes.
We need 2 glass plates, a casting frame and a casting stand. Place the short plate in front of the plate like that and then put it into the casting frame. After putting the casting frame, we must secure it by clamping it, and after we have assembled the apparatus, we need to take the comb and we place it in between the glass plates. We take a marker and make a mark approximately 1 centimeter below the comb and check for leaks with water before pouring gel. As you can see right now, the apparatus is not leaky, so therefore we'll draw out the water using a piece of filter paper. Now we'll be casting the resolving gel and pour the gel till the gel is at the same level as the mark. And then we remove the bubbles by adding a layer of isopropanoate on top of the gel like that. After the resolving gel has polymerized, we will try out the isopropanoate using the filter paper.
Next, sample will be loaded into the well that are numbered between 1 to 10. A lemon sample buffer will be used to denature the protein, and it contains glycerol, making it denser than water so it will sink to the bottom of the lane. To load the gel, a special tip must be used for the micropipette. It must be a very slender tip so it can accurately deliver the sample where it is supposed to go into the gel. Then, carefully loaded 20 microliter of sample using 20 microliter of micropipette to prevent from collecting air bubble. Slowly, the sample was loaded into the well located between the 2 gel plates. The loading dye is denser than water, so it should be sink to the bottom of the well. Common error when doing this step are missing the well when injecting the sample. Besides, badly prepared sample may contain air bubble, making it difficult to pipette. This causes insufficient sample being loaded into the well. Loading sample very quickly will also result in insufficient amount being loaded, making it overspill from the well. If the sample did not stay at the bottom of the lane, do not continue this step. Instead, you align the pipette tip at the 1 mm gap separating the 2 plate glass and slowly re-inject it again. This time, it will land at the bottom of the lane correctly.
After the sample application procedure, an electric field is applied across the gel, causing the negatively charged proteins to migrate across the gel away from the negative electrode and towards the positive electrode. Also, small proteins migrate relatively easily through the mesh of the gel, while larger proteins are more likely to be retained and thereby migrate more slowly through the gel. The stacking gel is used to ensure that all of the proteins arrive at the separating gel at the same time because gel wells are around 1 cm deep. So, in the absence of a stacking gel, proteins would all enter the separating gel at different times, resulting in very cement bands. As we have seen previously, the running buffer consists of glycine and chloride ions. So, in the sample wells, we will have proteins, chloride ions, and lysine. Proteins and chloride ions are negatively charged. Well, glycine can exist in 3 different charges states, depending on the pH. The running buffer pH is equal 8.3, which above the pHi of lysine. So, at this pH, glycine is negatively charged. Once the electric current is applied, proteins, chloride ions, and lysine are forced to enter the stacking gel, where the pH is equal 6.8. At this pH, glycine switches predominantly to this metallic neutrally charged state. This loss of charge causes them to move very slowly in the electric field. The chloride ions, on the other hand, move much more quickly. Chloride ions migrate in front of the proteins as leading ions, and lysine molecules migrate behind the proteins as initial trailing ions. Consequently, proteins are trapped and concentrated into a narrow zone between the chloride ions and glycine. This position carries on until it hits the separating gel, where the pH switches to 8.8. At this pH, the glycine molecules are mostly negatively charged and can migrate much faster than the proteins. Proteins are separated, so higher molecular weight proteins move more slowly through the porous acrylamide gel than lower molecular weight proteins. Due to the relatively small molecule size of brown, pink, and blue, it migrates faster than proteins. And by optical control of the migrating colored band, the electrophoresis can be stopped before the dye and before the sample have completely migrated through the gel and leave it. At the end of the electrophoretic separation, all proteins are sorted by size and can be analyzed by other methods, for example, protein staining methods and immunological detection such as the Western blot technique.
Today I will talk about how to stain gel using pH blue. Firstly, fill the microwave container with a distillate water. Rinse the glass plates because water helps to lubricate the plate and make them easier to separate. Then, carefully remove the stacking gel. Next, put the plate upside down so that the water will get between the gel and the plate. Therefore, the gel will come right off into the container. Next, the gel needs to be washed for a few times with a distillate water, but make sure to put the water in gently so it doesn't break the gel. Fill the container with a distillate water and microwave it for about a minute and let it shake on a shaker for about 5 minutes. Repeat the wash 1 or 2 more times to remove all the SDS, then transfer to the pH blue. Before transfer the gel into the dye, make sure the excess water was removed because we don't want to dilute the dye. Then, we microwave the gel about 30 seconds. By heating the gel again, help the dye to be absorbed into the gel and then put on a shaker again for about 10 minutes. Next, remove the excess dye from the gel and put into the distillate water. Rinse the gel with distillate water for a few times. Then shake in water overnight or until the stain is complete. After destaining, the protein appears as a blue band against a clear gel background. Typically, band of protein can be visualized. Another method of staining involves using Comasi stain. First, place the gel directly into the Comasi stain for about 20 to 60 minutes. After staining, transfer the gel into the staining solution made of methanol and acetic acid. This protocol does not require microwave. Silver staining is a more sensitive method than Comasi staining, capable of detecting 2 to 5 nanograms of protein per band on a gel. First, fix the gel in a methanol acetic acid solution. Then, oxidize the gel by placing it in an oxidizing solution. After this step, wash the gel several times with distillate water before staining it with a silver nitrate for 20 minutes. Rinse the gel with water to remove excess silver nitrate. Next, develop the gel by immersing it in an sodium carbonate solution. To stop the development process, place the gel in an acetic acid solution.